So thank you so much for the intro, and thank you so much to the steering committee and the organizers for giving me this amazing opportunity. I'm a synthetic biologist by training, and during my postdoc, I use these engineering-inspired approaches to create precise gene therapy. And so today, after a brief introduction, I will deep dive into two such applications in the brain and in the retina. So let's go. So what is gene therapy? It is essentially the delivery of genetic material into cells for promoting health. It is usually comprised of a delivery vehicle. In the case of the central nervous system, the CNS, we usually use the adeno-associated viruses, or AAVs. And inside the capsid of the virus, we have the genetic material, which encodes for a promoter that drives expression of the cargo. And the cargo can be a healthy copy of a gene that we would like to target to deliver to patients with a mutation. It can be the CRISPR regulation machinery. It can be miRNA, siRNA inhibition, and so many more modules. And so over the years, advancement in the field included two major avenues: in efficiency, essentially reaching high enough expression levels of our cargo in the target cells. And here we can improve the delivery method. For example, in this beautiful image, where we see GFP in the brain after nasal injections combined with ultrasound. So super cool. However, high expression is not always the best when we talk about the CNS, as sometimes we have neurotoxicity due to prolonged transient expression. And so we need to create a way to do distinct levels or distinct expression levels in all cells, and hopefully also some sort of temporal control to limit the time frame of expression. The second avenue is specificity, essentially expressing it in the right cells to avoid off-target effects. And here we have the promoters or the enhancers that have been talked about in the first session. So essentially we have this, I can show an example where we have the GFAP promoter and the synapsin promoters expressing cherry and astrocytes and GFP in neurons. And this is a tremendously helpful approach. In the CNS, we know that there are highly diverse cell types and specializations, and so we need, or it requires, a very high resolution to be able to really target these neuronal subclasses. And you can see here a single-cell RNA sequencing of the brain, of the mouse brain, over 100 different subclasses, neuronal subclasses. And so to tackle such challenges, during my postdoc, I designed biomolecular circuits to target gene expression in the right amount, the right place, and in the right time. And I'm going to go over each one. And first, let's deep dive into the right amount. So going back to this beautiful image of the GFP in the brain, you can see very nice expression. But you can also see there's high variability between cells, coming from the fact that some cells received high viral titer, expressing more GFP, and some received less, expressing less. And so it is very difficult to control the expression levels of every single cells. And this becomes crucial when we talk about over 300 dosage-sensitive disorders in the brain. So what is dosage sensitive? So think Goldilocks, okay? If we take RI1 as a proof of concept, we have healthy people have 2 copies of a gene. Sick people have either 1 copy, causing the Smith-Magenik syndrome, or 3 copies causing the Potocki-Lupski syndrome. So we can try and do gene therapy. We can CRISPR activate the RI1 for SMS patients. Unfortunately, because of this variability in delivery, some cells might get too much of that expression, or too much of that CRISPR, and they will overshoot into the PTLS 3-copy regime. And so the idea is to create a more uniform regulation or delivery, so that we also have more a strict expression in all cells, as well as tunability. And we did that by creating an incoherent feed-forward control loop, or an IFFL, which is essentially, here we let me do laser pointer. So we include, instead of the dosage going directly to activation, we also have an inhibitory arm that can deactivate the activation or the gene of interest. And so what happens is that a cell that gets a high dosage, it will get a lot of the gene, but it will also get a high amount of the inhibitor. And these two will cross each other out, and you will have uniform gene activation in all cells. And here we use the CRISPR activation as the activation arm, and the protease that can deactivate it as the inhibitory arm. And now I will show you 2 data sets. First, we activated RI1 in cortical cultured neurons, mouse cortical neurons, where we deliver AAVs with either the CRISPR activation system, here in red, or with the IFFL, where we have the inhibitory arm as well. So we did microscopy and we imaged these single mRNA transcripts, and here we quantified them. You can see the percentage of the RI1 positive area and the normalized cell count as the y-axis. As you can see, with CRISPR activation, there's a very wide distribution. Here, there's also some cells that acquire or exhibit signal more than 8 or even 9-fold more from the untransduced median. When you look at the IFFL, you can see that this histogram is much narrower, and all these kind of overexpressing cells are much less populated. And so this was promising. So we did the next step. We did something very similar, but here we're targeting SMS patient-derived cell lines. And here you can see the SMS signal for RI1 as measured this time by flow cytometry. I'm also plotting the 2x SMS line and the 3x SMS line to kind of give you an idea of where we would like to be in terms of healthy amount. And so CRISPR activation, again, you can see it's basically sitting on the 3x line with one of the replicates completely here in this unwanted region. It's too high. But if you look at the IFFL, you can see that this is kind of shifted to the left. And also this region here is much, much less populated, which was nice to see. And so the next step is we're doing now experiments in mice models of autism, of SMS. Okay, so now I am moving on to the next project, which is expression in the right place. And we are zooming out of the brain, and this time into the retina, and targeting these retinal ganglial cells. And these cells are responsible for propagating all the visual stimuli to the brain, to the optic nerves, to the optic nerve, and they've been implicated in neuropathies. And so ideally, we would like to target them in vivo using gene therapy of growth promoting and repair genes. So we need to target these cells alone. Unfortunately, it's not as simple as that, as RGCs are actually made up of over 40 different subclasses, as you can see here in this RNA sequencing based clustering. So, and each one of these subclasses have very high variance in resilience to optic nerve crush, as can be seen here. And also, even more importantly, when we do gene therapy with growth-promoting genes, the same growth-promoting gene that can cause regeneration and repair in some of these subclasses might actually cause toxic effects and cell death in others. And so again, we need to have a very high resolution to be able to really target these cells, these subclasses individually. And we have not seen promoters or enhancers that can really target these, or at least not when we started before the first session today. Okay, so to overcome this challenge, I've used this technology that we've been developing in the Gao lab for the past few years, and it's called RADAR. And the idea is that we have RNA sensors that can express a protein of our choice as a function of a specific mRNA in the cell. And so this is how it works. We have the RNA sequence here. We have a cherry marker that's always on. And then we have a sensor sequence here with a stop codon. And the stop codon does not allow for the expression of the downstream gene, EGFP in this case, until our sensor meets this target mRNA that's expressed in cells, together with the ADAR protein. And ADAR is an RNA editing enzyme that's present in all cells, especially in neurons. And so what happens? So we have the sensor sequence binds the target mRNA, creating this double-stranded region that can recruit the ADAR protein. And now ADAR is able to edit the A in the stop codon into an I. And now there's no longer a stop codon, and the downstream gene can be expressed. And so GFP here, the overall GFP expression is completely dependent on the presence of the target mRNA. And so I developed this technology to target retinal ganglial cells as a first step. And then the second step is to target just the W3B subclass, specifically out of all the different subclasses. So I'm showing you just one data set. I don't have time, where we used the radar system, we used the sensor, we injected mice in the retina via intravitreal injections. And here are the results. You can see the ganglial cell layer here in blue, as denoted by the RBPMS positive. This is an RGC-only gene. And you can see the cherry here is kind of throughout the layers, really nicely expressed throughout the layers. But the sensor output, which is supposed to be only specifically for our BPMS cells, you can see it is really nicely correlated with the ganglia layer. So we have the efficiency is not that great, but still specificity is really nice, correlated again with the ganglia layer. So that was again promising. And so I have another project at the right time that I've started working on. Unfortunately, I don't have time to talk about this. No time for the right time. But I would love to talk about this offline with anybody who's interested. In terms of future plans, maybe one sentence, I would like to continue developing my tools to promote neural health and repair, and perhaps even combine these tools to create quantitative, temporal, and spatial control over gene therapy. If we take the retina as an example, imagine a way we can target each of these subclasses with your optimal growth factor gene and also control it in terms of dosage and the right time frame of expression. So that's it for me. Thank you so much to my advisor, Xiaojing Gao, to my collaborators for the beautiful images, to my co-authors, my funding sources, and thank you so much for listening.